From Microbe TV, this is Immune, episode number 96, recorded on August 19th, 2025. I'm Vincent Racaniello, and you're listening to the podcast about the body's defenders against disease. Joining me today from Ithaca, New York, Cindy Leifer.
Hi, welcome back.
Is it getting cool up there like it is here?
Oh, it was delightful last night. It was great sleeping weather. It's been kind of hot the past couple days, but it cooled off last night.
Yeah, here it's cooled off. We got this hurricane coming in.
I was reading massive waves. Is that?
Yeah, but I think we're supposed to get rain Wednesday and Thursday, and that's it. So it doesn't seem to be too disruptive. Also joining us from Cleveland, Ohio, Steph Langle.
Hello. Hi, everyone. Great to be here. Yeah, a little cooler. The undergrads are back on campus. There's quite a different energy, also different traffic patterns when the undergrads come back.
Yeah.
So, yeah.
And from Madison, New Jersey, Breanne Barker.
Hi, great to be here. We do not yet have our undergrads back. They come back on Friday. So I have a couple more days of quiet before it'll really ramp up.
Yeah.
Yeah, I'm on the other side of that undergrads. Thursday, we drive my younger one to college. So it's very exciting.
Oh, that is neat. That is the other side of things, right? Different feelings, different vibe. That's great.
Well, it's going to be warm again. Don't worry. It always does, right? In September. Sometimes even into October it gets warm, although not that high humidity stuff that we already had. That was July. If you enjoy these science programs at Microbe TV, we have a bunch of them besides Immune. We have like 9, I think. And when we talk about science, we give you the truth. We don't lie to you about science. We don't give you disinformation. And if you like that and you enjoy it, we'd love your support. We don't do advertising. We depend on listener contributions, microbe.tv/contribute. And if you're on Patreon, we now have a yearly subscription option. And if you do that, you get 10% off. So maybe you just want to have your credit card charged once a year. Maybe you want to feel like you're a dedicated helper for a year. That would be great. It's just a convenience thing. We know you're going to be here every month forever anyway. But if you want to have fewer charges on your statement, your monthly statement, go over to Patreon.
And full disclosure, we don't get paid to do this. We volunteer our time. The money goes to keep the lights on and to keep the videos flowing and to help out with the behind the scenes support so we can be here to do our thing.
We have a staff here at Microbe TV. We have a full-time person. We have a bunch of contractors, thank you. And they do things like social media and editing. Yeah. Because with 9 podcasts, you need some help.
I do remember the years before maybe you had some of this help, Vincent, and it was a lot for you to do. And as the number of podcasts kept increasing, there was obviously a time when it had to.
So it's an interesting story. I did all the editing and posting, recording, editing, and posting myself for many years. And my lab shrunk over those years because I couldn't do both. You cannot do intense SciComm and have a lab. You can do moderate SciComm and have a lab. But for some reason, I felt it was my calling to do this. And I had a long lab career, and I thought that would be okay.
Mm-hmm. We're glad you did because we need it now more than ever.
Yeah, it's more than okay.
Yeah. And so then the COVID came along, and we became a nonprofit because all of a sudden people were donating. You know, I'd put these donation things up years ago, and hardly anyone donated. And then suddenly, bam, lots of donations. Mm-hmm. So we became a nonprofit. We got this facility, the incubator. And people to help us. Mm-hmm. So, you know, we're real. We are a physical entity. You can, in fact, visit us if you want. Just email us if you want to do that. But anyway, today we have 2 very interesting papers for you. You're going to love it. So stay with us. Don't go. And we're going to start with a snippet from Steph.
Sure. Okay, so the paper that I'm going to snippetize today Although I sometimes make it a little bit longer, but I'll try to keep it a snippet. But it's in Nature Biomedical Engineering, and it's called Floss-Based Vaccination Targets the Gingival Sulcus for Mucosal and Systemic Immunization. The first author is Rohan Ingrel. The last author is Harvinder Singh Gill. And these investigators are at the University, Texas Tech University, North Carolina State University, and University of North Carolina at Chapel Hill. So the premise of this paper is that people are very interested in pursuing non-injectable vaccines to engage mucosal sites and a mucosal immunity as well as systemic immunity. So to give you an idea of why those two might be different, viruses infect epithelial cells and other cells mostly at mucosal sites, the respiratory tract, the gastrointestinal tract, the reproductive tract. And there are a lot of immune cells sitting right below the single layer of epithelium that really are coating all of your mucosal spaces. Below there, you have many innate immune cells. You have macrophages, different types of innate like cells, ILCs. You also have adaptive immune cells, B and T cells, that coordinate their movements to go to germinal centers that sit in the gut. It could be Peyer's patches, the mesenteric lymph node. We talked about in the upper respiratory tract in a previous episode that there are lymphoid structures that allow cells to travel to the nasal turbinates and back, and of course the lung. And these sites allow for engagement of both IgG and IgA. IgA is produced in huge quantities at all of our mucosal sites, but both of these antibodies are working in tandem to protect the surfaces, and that really is the first place of infection. Now, it's the case that most of our vaccines are injectable they are delivering antigen to systemic lymph nodes to engage germinal center interactions that promote very potent affinity matured, mostly IgG responses. And those are associated with protection against severe disease. For SARS-CoV-2, we talked about it. These IgG antibodies are moving into the lung. The lung definitely has a lot of blood going to it. That's where IgG can interact with the virus. But what if you could engage both? Because if you could prevent replication, you can prevent further seeding into the lung if you could stop it in the upper respiratory tract, or transmission to others. And so being able to engage, I joke that, this has been something we've talked about on this show for years. It's possible. It's just tricky for many reasons. How do you engage mucosal sites in a very diverse population with a diverse mucosal health? People eat different things. They have different microbiomes. There's different pollution in the environment. They may smoke, they may not smoke. So it is a bit more consistent to give an intramuscular vaccine only because that's how we know how to measure the immune response appropriately. So we can pull blood, we can get a titer, which in the case of influenza, which we'll talk about, is associated with a protection level based on an assay. Mucosal samples are more tricky. You have to deal with mucus. And what are you looking for, IgG? Or are you looking for IgA? And so a lot of groups are working on these types of questions. And this group was really focused on this idea of non-injectable vaccines.
Yeah. One other thing that I always think about when I think about specifically GI immunity is that in some situations we end up with oral tolerance. And so some antigens that we encounter through the gastrointestinal tract are things that we end up not making a strong immune response to, or making a different type of response, so that we don't have pathology. And, you can think about all of the antigens in your food as things you don't particularly want to have a response against. I, at least, this is where I'm lucky to have co-hosts, do not really understand how that happens. And so I think that one other thing I've always wondered about is, how do you set up a oral vaccine or a mucosal vaccine and make sure that it doesn't lead to a tolerance response, because that would actually be particularly problematic. And how do we control sort of the difference between those types of responses?
Yes, that is very true. It would not be ideal for us as a species if we were having anaphylactic shock every time we ate food or came across a commensal microbe. And so we do have this tolerance mechanism you talked about. The way to get around that, and why just giving protein in the nose or swallowing recombinant antigen probably won't work, is you need some way to tell the immune system that, hey, you need to react. I want you to respond. So we probably would add an adjuvant to that protein vaccine to stimulate a toll-like receptor in the mucosal space to cause a mini inflammatory response so that those cytokines could tell the cells, hey, come here, there's antigen. Or you attenuate the virus and make it an attenuated viral vaccine. Rotavirus and poliovirus are the two approved, as well as influenza has a attenuated, it's called FluMist. It only works in kids, not adults, because of pre-existing immunity. But that is how you would tell those mucosal sites, hey, you need to pay attention, is some type of mini inflammatory event, which would be the virus is replicating but not causing problems, or I'm giving an adjuvant, BCG or CPG or Adavax is a TLR7/8 adjuvant. Yeah.
I think there's one little thing that I want to add to that, and that is when you inject something into a muscle, you're causing some muscle damage, so you're inherently going to induce some of those danger-associated molecular patterns, which act as those innate adjuvants, inherent adjuvants. And if you add an adjuvant to an injectable, it will mostly stay there, right? So you're injecting it into a muscle, and it's going to mostly be in that muscle, that liquid. When you take something orally, it diffuses really easily. And if you try to mix together into one formulation, a pathogen-associated molecular pattern, or a MAMP or a DAMP or PAMP, and the thing you want to generate an immune response to is you put it in your mouth and it's just dissolved in saliva and they go different places. So I think there's actually a fundamental thing that needs to be done differently, and is that it would normally be conjugated or have some other mechanism of being able to elicit that response that you're talking about. Because I don't think it's just, like you said, you can't just eat whatever it is, because we're designed to be tolerant to that. But at the same time, you just can't throw a bunch of microbial structures in there and expect it to work, because in essence, you're also eating microbial structures whenever you eat too, right? Because whether you're eating harmless bacteria or you're actually eating like LPS that's come off bacteria in the food preparations or whatever, it's already there. So there's something fundamentally different that we need to do about mucosal vaccines. Yeah,
and I think the point that Cindy makes about the mechanical damage that happens with the injection, and that leading to some of that inflammatory process, is related to something that I was kind of thinking about with this paper. So, that's actually why I brought all of this up, because that was a point I'm thinking
floss,
about.
How many of us? First of all, how many of us floss every single day like we're supposed to?
I will say I became kind of obsessed with it. I'm one of these weird people. I will. Like, you catch me, I'm probably flossing. I do love it, but it took me a while to get there. So I'm not wasn't always perfect.
But we'll circle back around to this, Brianne. But I'm imagining that what you're envisioning is many people who don't on a regular basis, when they go to floss, there's a lot of bleeding and irritation of gums.
Well, I will tell you my thoughts on that, because yes, I'm thinking in that direction as we get there.
Yes. Yeah. And many companies are working on this issue of where do you deliver it, to what bug, how are you going to stimulate it? Are you going to conjugate it? Are you going to enterically coat in a pill to make it go to the stomach? So many different strategies. And the strategy that this group came up with, which honestly, before this paper, I was not thinking about, is a floss-based vaccine. So this is vaccine antigen that has been coated on floss thread, and in these experiments used to floss the 2 teeth of a mouse. And where they're trying to deliver this antigen is in some tissues that I don't often think about, is the buccal and sublingual tissues. They are unique because at the base of something called the gingival sulcus, they have this junctional epithelium that is non-keratinized epithelium. So it has high permeability. There are abundant amount of immune cells there. And this kind of natural leakiness, quote unquote, allows for antigen entry into what seems like the systemic, your blood, and possibly the mucosal tissues at that site. And so to exploit this area, which if you read interviews of the lead author, I think he was reading an article, just reading off topic, and learned about the gingival sulcus from a completely different field and thought, he's a vaccine engineer, and thought I could deliver an antigen there. And so in this paper, they exploited that. They proposed this vaccine coatal dental floss method. And they show in these neat pictures how they coat it, that when you have braided thread, the antigen gets stuck in the gaps, which is not ideal. So they had to use this flat tape kind of thread so that you can get even delivery of the antigen. And they give a nice demonstration that the junctional epithelium they're trying to get to is like at the bottom of where the tooth and the tissue lie. You have to get down in there, and they deliver this antigen to the mouse. And they show some little pictures of their two front teeth, if you can envision what the mouse teeth look like. And they show delivery of the antigen in the mouse. And so
this panel B with the mouse, it looks like the mouse is flossing itself.
Yeah,
got that stick there drawn in.
It does, the way that it's...
Yeah, it does. That's so
I wonder
funny.
if you have to anesthetize the mice to do this. I mean, I guess you could hold their head really tightly. Yeah, you could hold them tightly.
I don't know. I think I'd want them knocked out to do this.
They can be very frisky.
Well, so one of the things that I wondered, and I didn't look in the details of this. I mean, when we floss, you put it between the tooth and you go back and forth, right? Or at least you go in and you pull it out. So do they put it in there and just let it sit to allow the vaccine that's on the tape to diffuse off and get there? Or is just putting it in between and then pulling it out, is that enough to deliver enough of the material to generate this vaccine response?
I mean, I touch my gum when I do that, right?
Yeah.
And go up and down. They told me to go up and down on either side. Yeah. Right. So in that case, if you're doing that with the mouse, are you touching the gum? How many times you go up and down on either side? You got to standardize.
There would be a lot of variability in the delivery. Yeah.
So I looked in the method section. The mice were anesthetized, and they actually were not allowed to have food or water for an hour after flossing. Mm-hmm. Also, they bought the floss at CVS.
I love
that. Well, if they bought it at Fisher, it would be $100 a roll. So that's good.
Right. Be upcharged out the wazoo. If you go, so I put a link in the show notes, and hopefully we can include this. It's the Science News report of this paper, and there's a video, and the video shows you exactly on a very small scale the mouse's where they're trying to deliver this. And it's not as simple as just touching the gum. They are actually getting down, kind of how we're probably supposed to floss, but they're getting down into that crevice, which we'll talk about in the human data they present, because I think you bring up a great point that user error and actually getting down to where the junctional epithelium are is going to be key for consistent delivery.
Yes.
That's a great
The floss
video.
was pulled into the space and performed with a back and forth motion 10 times.
They sawed it. Yeah.
They sawed it.
No, they did. They went.
Really...
So when I floss, I put it in, and you're supposed to like pull it against your tooth and then pull down, because really the bacteria
maybe you should
form a
demonstrate
biofilm.
for us, Cindy.
I might have floss. I'm not going to floss my teeth on video. But you're supposed to pull it, because the idea is that bacteria form biofilm on your teeth, and you just want to break that biofilm. That's the whole goal. But this is really putting it down, because you need to. They saw it back. I'm watching this video. They saw it back and forth in that, and it bleeds, and I think a little bit. That seems a little rough.
Know.
But
is that blood, or I think that's the fluorescent?
It might be the fluorescent dye.
Okay, so here's my question.
It looks pink to me.
All right,
because I think that we all, are trying to imagine flossing a mouse, and this being very foreign to us. So is flossing a mouse a model of a person with bad oral hygiene? Because the mice probably have never been flossed before and don't brush their teeth? Or is it a model of people with good oral hygiene?
Well, no, that's a good point, because if there was a release of blood, actually that might help your antigen uptake.
Right. No, this is why
this
I
is
got
what
on
you're
that
wondering.
whole track.
Right. But then if you have bad oral hygiene and you have more bacteria, more biofilms, you'd have less penetration possibly. That's right. I think these are all really, interesting things that this group should study is in models of, a lot of adults have gum disease, clinical gum disease. So that would be a challenge. But yeah, to check out this video, it is just really interesting. And they went on a series of experiments where now they want to understand the immune response, to different concentrations of their vaccine, with and without an adjuvant. And as we could expect, if you add an adjuvant CpG coating along with the protein, you have higher responses, both in the serum, but particularly in the mucosal spaces. This makes sense as, again, we're trying to break tolerance to engage those mucosal responses. They did a variety of doses to try to understand what dose, how many times. And in a challenge model against influenza, they do see protection, and they actually see enhanced protection over an intranasal vaccine, which, just as a caveat, the mouse model of influenza, can be a terminal model, meaning they do die to the infection. And so this is a model of probably systemic protection. You're increasing systemic IgG. They did not do kind of the transmission studies to understand mucosal protection. So they find that this method was sufficient to induce the appropriate immune responses. They look at CD4, CD8 immune responses to different proteins, then different cytokines were secreted. So it's really seems like they are able to deliver the antigen, engage the immune response to a degree that provides protection in the mouse. And they wrap it up with understanding whether dental floss. I will say, let me go back to the mouse study. I think they kind of buried this data a bit, but I was interested in IgA, and it didn't seem like the dental floss really increased salivary IgA, which was interesting. I would have assumed it would have, but I think, again, this is more delivering antigen past these tight junctions to the blood, to the systemic.
That's what I was thinking, is that this
like these cells up here.
Kind of like a systemic delivery. Mm-hmm. Mm-hmm. And it might only be a systemic delivery because they're causing that damage.
A little trauma.
Little trauma, micro trauma. So they wanted to see if they could deliver the antigen to humans. And so they did this same, they did a commercially available dental floss pick. They allowed healthy volunteers ages 18 to 50 to use the dental floss pick, and they coated it with food dye, so blued food coloring to enhance, visibility. And what they found was 59% of the 100 microgram coated floss was delivered to the gingival sulcus, while there was some still remaining to the floss. And so I think 56% of the participants flossed between 2 and 11 times, with 5 passes being the most common. And I don't think they were able to let me see, were they able to distinguish which was more efficient? I think they just reported that as that's how many passes people did. But they were able to see delivery, but not in 100% of the volunteers. And I think this is going to be the challenge with applying this to human population, is user error, differences in oral biology and physiology, but really neat. And I think, gosh, if I could get my vaccine just delivered to me by Amazon that sits in my, or maybe in the fridge or in my medicine cabinet, and I get to do it when I want. It's a pretty neat concept.
I like that they did these surveys of the people afterwards, and they said 67% of the people said they would be more inclined to receive flu or coronavirus vaccines by floss. I bet this would really reduce anti-vax sentiment.
I mean, even more than taking a pill, right? Because there's, people don't really, especially kids, it's hard to take a pill. It's easier than an injection, but this would be the most easy. If it could be, if people could deliver it effectively? I'm envisioning like a little packet where you have pictures of how do you do it. Yeah.
I guess the biggest concern would be if you're trying to regulate something and approve it, not knowing exactly how much you're going to deliver.
Yeah.
And the consistency could be a real challenge.
Yep.
Because if it's not done properly and you're not protected, and you think you're protected, I think there's some significant challenges with actually implementing this, but I really love the creativity of it.
Love the creativity. Yeah. And I agree. I think if you're not getting this administered in the hospital or I'm sorry, in like an outpatient setting, okay, so where is it being stored? How long did you let it sit there? Does the antigen, I'm sure the antigen is going to degrade over time. So it is difficult to implement it and get it approved. Cindy, you're right. But really neat kind of creative work.
They make this interesting statement in the discussion. It can only be used for individuals with teeth.
I know.
Right,
I read that. I laughed
Babies
out loud when I read that. I mean, like, what the...
So kids, right, up to 6 months, you don't have teeth. But
or elderly who
if
have
they're
dentures.
Really old. What if you have dentures? Then you can't use a denture, right?
No.
Right.
This would not be for you.
Yeah, it's interesting. Obviously, that eliminates a lot of older people who need flu and RSV and all that, COVID vaccines.
Right.
So not sure what's going to happen, but it's really curious that they explore everything, right?
Mm-hmm.
I like
it's cool
that very
to think
much.
about.
Yeah.
So this made me find a Frank Zappa quote because you remember there's a song of his called Montana. I might be moving to Montana soon just to raise me up a crop of dental floss, raising it up, waxing it down in a little white box that I can sell uptown.
I might be moving to Montana soon just to raise me up a crop of dental floss.
You should hear Kathy Spindler say that. She's got it down.
I've never heard that.
I have not heard that either.
You never heard that song?
No.
No, I need to get into my Frank Zappa era. I have not experienced Frank Zappa.
Well, you're a bit young for Frank Zappa, but he's a very smart guy. He was. Have you heard any? There's a whole page of his quotes.
Yeah, I was going to say, I think this was discussed a little bit on that one TWIV episode, and we were on a page of quotes.
There's the one about the percentage of particles in the universe. 5% neutrons, 5% protons, 5% electrons, and 75% morons.
It's a positive way to think of it.
It's a lot, but I like the dental floss.
Yeah.
Thank you, Steph.
Sure.
So we're going to stay in mucosal immunity and secretory IgA now. We have a paper from Proceedings of the National Academy of Sciences. Human milk IgA promotes normal immune development by limiting Th17 inducing Erysipelatoclostridium ramosum in the infant gut. Erysipelatoclostridium, two different words. And I thought Steph would like this one.
I know. To our audience, I did not pick or suggest this for Vincent.
No, I just picked it because it has bacteria, and I would feel very insecure just doing an immunology paper if it didn't have bacteria or viruses in it, even though I have a wonderful support here. This is from, where's here we
Canada.
It's from Canada. Where's the darn things? I don't even know where the affiliations are. Are they all the way at the end?
Oh, that's a good question. Yeah, I didn't even look at that.
I'm on the PNAS website.
They're on the top on the website, and you have to click to get to them.
I have the PDF anyway.
It's Department of Microbiology and Immunology, University of British Columbia.
Mm-hmm.
I don't have my glasses.
UC Davis,
University of Manitoba.
Yeah.
Oh, page 11.
I think I think the senior author is at British Columbia.
University of Alberta.
There's a lot
University
of
Kuala Lumpur.
A lot of sites here, yeah. Well, I don't know.
Brett Finley's the senior author. And
yes. First author is Catherine Donald. Yeah. This is a cool study looking at secretory IgA in the gut microbiome. Secretory IgA is the most abundant antibody in the gut, and it interacts with the microbiome. It can help, it could promote or inhibit members of the gut microbiome. And in the first year of life, it's crucial to establish your gut microbiota, which in turn helps train the maturing immune system. Now, of course, in the first year of life, part of the first year of life, babies can't make a lot of secretory IgA, so they should get a lot of it through breastfeeding. And the secretory IgA in breast milk is made by plasma cells that are from the mother's gut. You know, we talk about the brain-gut axis, so here we have a gut mammary gland axis as well. It's just amazing how things communicate long distance. Of course, we knew that from the nervous system already, right? So they originate in the mother's gut, and they think that these secretory IgA antibodies target the gut microbiota in the mother and also in the baby. So, WHO says you should breastfeed for the first 6 months of life, which of course is challenging in many places. But, one outcome of this paper is that maybe we could find ways to make breast milk, which could help a lot of people who can't do it.
I just love what evolution did with the gut mammary axis, because it said, hey, the infant is going to be ingesting this fluid. At the same time, it has to contend with all these new microbes. I'm going to send cells all the way from the gut to the mammary gland that produce antibodies against the same bugs that are going to colonize the baby because they colonize mom and they're in the same environment. Yeah. It's such a neat.
And this happens in all mammals, right? Mammary glands. So this is a very successful strategy, obviously. So breastfeeding first 6 months, because it protects against many diseases, right? Allergies, asthma, inflammatory bowel disease, necrotizing enterocolitis and infections, and it promotes a healthy gut microbiota as well. So both the immune components of milk and other parts of milk control, they limit premature diversification and expansion of certain bacteria in early life, but we don't really know what controls what very well. And so that's one of the reasons for this paper. So at secretory IgA, this is something I didn't know, is 1 to 8 grams per liter of human milk, largest proportion of immune proteins passed over. And so it obviously is part of the passive immunity that the mom gives to the baby. For all of her infectious experience is going to the baby until the baby can develop their own antibodies.
That's why a number of vaccines are given during pregnancy, so that the mom boosts the production of those antibodies so that they can be transferred to the baby.
Right. Yep. So this group has previously worked with secretory IgA in mice, which you'll see are very different from humans. Well, we already knew that, but specifically, the topic of this paper is so the milk from mice limits segmented filamentous bacteria in the gut. It's also called Candidatus arthromyces. It's a candidate genus for this SFB. Without maternal secretory IgA in the mouse, these bacteria overgrow, and then you get Th17 responses that continue through adulthood, and you get more inflammatory disease in, for example, mouse models of asthma. So they're important for setting the immune system at a certain level. So these SFB are a key driver of Th17 cell development. And in mouse models of IBD and asthma, SFB exacerbate those diseases. But we don't have SFB in us. We've talked about this before. So what do we have? And that's part of the object of this study, is to find out if there's some bacteria in the human gut that has such a high impact on the gut microbiome and immune regulation. So they take advantage of a child cohort study that had been previously done. This is to look at people who are atopic, right, prone to allergic diseases. And so they have 300 participants, and they have a whole. These are all from Canada, different cities in Canada. They have a lot of information about features like whether antibiotics were used, the delivery mode of the babies, whether the mother smoked, whether the mom had atopy, and the age of the mom, and the race or the ethnicity. And so they have milk samples from these. And so they took this milk IgA, and they have a series of bacteria. They have a panel of bacteria that are known to be in the infant gut, that are commonly found in the infant gut. And they ask, do the secretory IgA antibodies in the milk from these mothers who are in this study, so they have mothers with and without atopy, do they bind? What bacteria do they bind? So they do ELISAs essentially, and they identify, binding to various bacteria. Some of the results are quite interesting. So Bacteroideaceae and Bifidobacteriaceae were bound at the lowest level by SIgA, and these are thought to be beneficial in early life. So that makes sense, right? You don't want to target them. On the other hand, targeting them may not be a bad thing, as you will see. Mm-hmm. Enterobacteriaceae species typically decline in the first months. They have the highest targeting by secretory IgA, and that's also found in human species and adult humans as well. And basically, the binding of human milk secretory IgA is similar to what's been previously found in human adult fecal secretory IgA. And that's, again, because the B cells originate in the gut. So the total quantities of secretory IgA didn't differ between atopic and non-atopic mothers. But total milk secretory IgA was negatively associated with several immune markers, including cytokines that are known to drive neutrophil and Th17 responses, leukocyte infiltration, which makes them think that human milk secretory IgA targets and limits Th17-inducing microbes in the infant intestine, which is kind of what the SFB the SFB does, right? So then they did metagenomic sequencing of these samples, and they get 5 species of bacteria that have strong associations with the level of secretory IgA in the meat. These are Bacteroides, Bifidobacteria, Bacteroides fragilis, Bifidobacteria, Bifidum Bacteroides uniformis, Bifidobacterium brevin E. ramosum. So E. ramosum is the one in the title. We're going to just call it E because it's too long to keep saying over.
Yeah,
and
I was
over.
impressed you couldn't pronounce that.
I practice it.
Yeah.
Clostridiaceae is easy. It's the first one, Erysipetalus, and I just never get it right.
You did pretty good, though.
Yeah.
So they found that basically secretory IgA binding patterns are more indicative of the gut microbiota composition than are the total levels.
And this is the gut in the infant.
The infant gut,
with the IgA that came from mom.
Right. So of all these...
So there's a little caveat to that, in that they took the antibodies from the mom's milk,
right,
and they looked for binding to the bacteria that they grew in vitro. Yeah. So there was some difference. They weren't directly from the babies.
No, that's right.
They did talk about that in the discussion, that was a limitation to the study, because sometimes there's some changes when you grow bacteria in vitro, and something that binds in vitro may not bind in vivo, but they didn't seem to think that was probably going to be much of a problem.
Right. Well, in here, they are using the metagenomics from what's in the baby.
Yeah. Yes. Yeah. And correlating it with the mom care. You're right.
So they didn't have fecal samples from the mothers. But they have this table, which is a list of common infant bacteria in the gut, and then they do metagenomics of the baby fecal samples to see how they correlate with the antibodies for those particular bacteria, right? So basically, E. ramosum was the only species in the infant gut that has a significant negative association with secretory IgA targeting. So they're saying maybe this is because secretory IgA limits this species in the human gut. And then they're going to do experiments to address that hypothesis. So they focused on E. ramosum because of that preliminary data. Now, so they have the fecal samples from these babies were collected at, quote, 3 months, end quote, but it actually ranged from 2.8 to 7.5 because you know how life is, right?
Yeah.
Sorry, I just couldn't do it on time.
Right.
I don't care if your NIH grant depends on it. But then they say, this way we have a time course. So E. ramosum was the top age-associated species in the infant gut. They have higher levels in older infants. It's also much higher in 1-year-old infants than in 3-month-old infants across this whole cohort, no matter whether they were atopic or not. And this is not new because previous studies have established correlations between E. ramosum and allergic disease from different cohorts as well. But what they find here that's unusual in the child study, the levels trend lower in feces of allergic infants than of control infants at the 3-month time. And then they catch up. So this illustrates a theme in this paper. That it's not the gut microbiota is not an all or none thing. There's a development in that first year. So there's a dynamic that has to happen in a certain way, right? Mm-hmm. So certain species have to come first, and then others. It's not just a matter of being there or not.
Right. Well, and this timing is important because at 3 months, you are still in that WHO recommended breastfeeding time. And at 1 year, you are after that. And so being able to see these kinds of differences at these particular time points also makes you think that this might be somewhat related to breast milk.
Right. So also E. ramosum were higher in exclusively breastfed infants compared to non-breastfed or partially breastfed infants, again supporting what they call a weaning-associated bloom of E. ramosum early in life. Same thing happens with SFB in mice.
So I wanted to ask you guys, what did you think about the data that they used to argue that?
The part of breastfed versus non-breastfed?
Yeah. So there's exclusive, partial, and none.
Yeah.
And there's quite a spread of the data. And I don't really understand where the significance is coming from. So they have an average bar that's way lower in the exclusive breastfed and way higher in the none. So that suggests that there's lower levels of this E. ramosum in the exclusively breastfed. But when you look at the scatter and the distribution of the dots that represent each individual, the data look the opposite to me. Now, I don't understand if there's just a bunch that are below detection limit and they're on the y-axis or on the x-axis, but the data don't seem to show what they are saying, and their data analysis doesn't seem to support that. So I felt like maybe I'm missing something, and I was wondering what you guys thought of that.
Yeah, that's a good point, because there are a lot of dots in the exclusive column, right? And most of them seem to be very high
above
up.
The average.
I don't know if but they shouldn't leave the dots out that are below the average. That wouldn't be right.
Correct. So I wasn't really sure what I was looking at and how to interpret that. So I kind of gave them the benefit of the doubt, but I am curious about those data.
I mean, I think the targeting of the bacteria that you don't want, and if you have more of those antibodies and you have less of that bug, that logically makes sense to me. But just looking at, and just to confirm, we're all looking at the same thing. Figure 2C is what we're...
In case you check the paper out. Yeah.
Right. In case you check the paper out. But it literally looks like it's flipped.
Yeah. So what I'm trying to say is that there's a scatter of dots from high level to low level. I don't want to belabor this too much. There's a scatter of dots from high level to low level. And then there's a defined average that they indicate. And the scatter is trending towards higher levels of the bacterium in the exclusive breastfed, but their average shows it is way lower.
Yeah,
and
it's
so
interesting.
it's very strange.
Yeah. I don't know what to, other than if they did have a bunch of stuff that was just zero, which wouldn't be the case, and if you can't log transform zero, so maybe it has something to do with that.
Yeah, maybe. Yeah. Anyway, I just thought that was curious, and I was wondering if you guys had looked at that and thought similar things.
So they conclude that the secretory IgA that binds E-ramosum are key to the breastfeeding associated differences, which, as you just heard, we're not convinced of from the data, from the plot anyway. Okay. Previously, others have found E-ramosum is a top age discriminatory taxon early in life. It increases after 3 months of age in healthy infants. They're also elevated in formula-fed infants. That's a different study. And a lack of antibodies, that secretory IgA antibodies that target E-ramosum is associated with later development of allergies. So again, the idea is that the secretory IgA is controlling the colonization of this bacterium in the infant gut. Also, in another study, E-ramosum was found to be a keystone member of the infant gut and a top allergy-associated microbe across the globe. So not just in Canada, but in many other countries as well. And so they are also identifying connections between E-ramosum and SIGA and breastfeeding status and allergic outcomes. Okay. This it's interesting that E-ramosum, besides being the target of secretory IgA, it also makes an IgA protease, which we've talked about bacteria making proteases that cleave antibodies before. I think that was a Steph paper. Right?
It was Immune 92. I looked it up because I was like, oh, that sounds really familiar. Yeah.
So that would suggest evading secretory IgA binding, right? So how does the secretory IgA work? How does it control the microbiota? One way would be to prevent the attachment bacteria to the intestinal epithelium, right? So they actually do an experiment to look at that. They have human-derived intestinal epithelial cells, and they add E-ramosum to those, and they see that it can adhere. These are CACO2 cells, they're called, and it's very hard to wash them off, so they're really stuck on there. And when the bacteria adhere, they induce cytoskeletal rearrangements and actin reorganization. So they don't just bind, they have a major effect on the cell. If they incubate the bacteria with secretory IgA from 20 human milk samples with very high binding, they reduce the adherence of E-ramosum to these intestinal epithelial cells.
Pretty cool.
Interestingly, not so interesting, if you heat denature the secretory IgA, it doesn't limit adherence. So you need to have the native antibody present. And also they purified secretory IgA from 20 milk samples that had low E-ramosum binding capacity, and that failed to limit adherence. So they're convinced that that's how one way that the antibody is working. Now, when they use epithelial cells that make a lot of mucus, the bacterium adheres very well, and it doesn't matter if you add the antibody to it. And maybe that's because the mucus is blocking somehow.
Are there differences in mucus amounts with age?
There,
like in the first year?
Yeah. So could that also be part of, changes in that microbe's colonization over time?
I think so. At least in the animals, it is much easier to deal with their intestines when they're younger. When in terms of processing, the older they get, the more enzymes we have to use to clear the mucus.
Yeah.
And the tight junctions get stronger and stronger. And yeah, there's more mucus production from goblet cells. So I don't know exactly in humans what the mucus structure changes are, but that was very interesting. And maybe why it does work so well in the infant, because maybe you don't have adult-like mucus yet. So
that's what I was thinking.
Yeah. Yeah. One thing I would love to know, and I think they could have addressed this maybe in just one experiment, but I'm curious how much of this is glycan dependent and how much is epitope specific. If you deglycosylate the antibodies with an enzyme to remove all the glycans in secretory IgA has more glycans than IgG. IgG has 1 FC, IgA has 4 FC glycans. The secretory component has like 9, and they interact with the bacteria themselves. If you deglycosylated it and you still saw the protective effect, then there is a B cell somewhere that produced this specific antibody for this bug. And I would love to know the dynamics of, is it glycans? Is it epitope specificity? Yeah. Interestingly,
E. ramosum did not adhere to murine intestinal cells. Of course, mouse don't have this bacteria, so if they did, it wouldn't stick. So we can't study this bacteria in mice. That's a downside.
So that speaks to whatever the receptor is that this bug is using to adhere itself does not exist in the mouse.
Yes.
Or is sufficiently different that it doesn't cross-react from mouse and human.
Yep.
Yeah.
So neat.
Interesting.
All right. So next, they say, okay, these cells, these epithelial cells, when we add E. ramosum, what happens? So they do RNA sequencing to find out, and then what they see is several inflammatory cytokines go up when you add. They call it an infection, but I don't know if that's the right word. They're just sticking to the outside, right?
Yeah.
Yeah.
Anyway, it doesn't matter. Heat-killed bacteria don't upregulate any of these inflammatory genes.
I find that part really interesting.
That you
have to have the live bacteria. The kill bacteria don't do it. So people have been studying kind of what we call vitapamps. Julie Blander's lab started that a while ago. But it's the idea that organisms that are metabolizing are creating something that lets the immune system know it's harmful because it's alive, right? Because if you have heat killed, it's not really dangerous to the body because it's dead. It's not going to replicate. Right. So there's something about being able to detect mRNAs or other things that are indicative of that current organism metabolizing can stimulate the immune system. And so I wonder whether some of that is involved in this, since the organism triggers it, but the heat-killed organism doesn't. But then you start to wonder if those are conserved across life, then why would it not trigger in mouse, but it does trigger in human? So I got stuck thinking about that, and I'm not sure I resolved that in my head.
But maybe with the mouse, you're not getting the attachment.
It just
so
gets
you
cleared
don't
out.
Necessarily get a chance to discriminate between those PAMPs.
That would suggest that maybe, I don't know, and I didn't look up whether they have a secretion system. So when they attach, do they inject anything into the cell? Because that might be a way that the cells could detect this live, because you're not going to have an injection of anything into a cell without attachment and without it metabolizing.
Yep. That could be. Anyway, these pathways that are other pathways induced all involve Th17 mediated inflammation, that like rheumatoid arthritis. So if you look at the genes whose expression goes up when you attach bacteria, they all fall into Th17 mediated inflammation pathways like rheumatoid arthritis, legionellosis, and IL-17 signaling. And if you add antibody, secretory IgA antibody, before this RNA-seq analysis, you don't get induction of these genes, right?
I did one of those microbiome sampling things a few years ago.
Oh, really?
And now I really want to go back and look at the data and see where E. ramosum is.
Yeah.
The problem is that I don't know how to know if the amount was a lot or a little. I also don't remember how to log in, but that's a separate issue.
That's a totally different issue for you.
Well, that's a common problem with everything.
Yeah, but I feel like I could look and see where it is if it's on the list, but I don't know how I would know if that was a lot or a little.
Well, did they give you read numbers or no?
I'd have to look back. It was quite a while ago. But maybe that's why I'm allergic to all the things.
Yeah.
Oh.
That would be really interesting.
Another experiment they did is to use Bacillus fragilis, which is known to be associated with milk SIGA. It adheres to these epithelial cells, but it does not upregulate the same inflammatory responses that the E. ramosum did. So adherence alone, again, is not enough to get it. So they looked at the genome of this bacterium. They compared E. ramosum with Bacillus fragilis, and they found a protein, fibronectin binding protein, in the E. ramosum genome that's not found in B. fragilis, which doesn't bind in the same way. So they're wondering whether this is responsible for the responses, right? As we were just saying, we were talking about an injection, but maybe fibronectin interaction. But I don't know why that would need a living bacterium. Though,
yeah,
right?
I don't know. That's yeah.
That's weird. Okay, so then they want to know what is the pathway from the binding to the turning up of these genes. So they use human-derived monocytic dendritic cells, so MODCs. They take the supernatant from E. ramosum infected human epithelial gut cells that we've been just talking about. This has got bacteria and cytokines in it. And they add it to these dendritic cells, and it drives differentiation towards Th17. All the markers that they see are Th17 related. IL-6 is another one that drives Th7 differentiation, also upregulated. TGF-beta, which is a regulator of Th17, is not induced. So that's consistent. And again, the cytokines as well as the bacteria are needed to induce this response. Because if you use supernatant of cells that were given heat-killed E. ramosum, it doesn't work. And finally, they take human peripheral blood-derived CD4 T cells. They expose those to infected MODCs, and this induces expression of IL-17, which is the cytokine made by Th17 cells, and heat-killed E. ramosum and IL-6 alone don't do that. So basically the epithelial response to live, and I could say live because it's a bacterium. I get nervous whenever I say live.
Yeah, I know.
The virologists are going to come after you.
Viruses. I'm going to go after myself.
You're going to come after yourself. Yeah, exactly.
Intestinal epithelial response to live and adherent bacteria drives a strong inflammatory Th17 response. All right. And again, this doesn't happen in mice. So what they did was to take germ-free mice, and they infect them with E. ramosum, and they quantify Th17 cells in the small intestine and colon, and they don't make a Th17 response, which is, we could have predicted that, right? Because it doesn't bind. So I mean, these germ-free mice have weird immune systems, right? So they took specific pathogen-free mice, which are not germ-free. So they have, I guess, more normal immune systems, right? And they treat them with vancomycin to deplete gut microbiome. And then they they feed them E. ramosum, still not induced.
Nada.
Th17 response is not induced in those either. So it's more complicated.
So do you think they started with mouse studies, or do you think that those were reviewer required experiments to say, come on, you have to test this in mouse because that way you can show this model?
It's been talking about this yesterday. It's always hard to know the sequence.
Yeah.
Because the sequence that's presented in the paper is always beautiful, right?
Right.
We did this, but usually so I don't know. if this was a reviewer 3 or what.
Yeah, my sense is the mouse studies came second only because this child cohort study has been going on in Canada for like 10 years now.
Yeah.
And so I think they're they have all this data and they're parsing through what are we seeing, and then they grab hold of something and test it maybe.
Yeah, poor graduate student had a project. Here, go show this works in mouse. And they're like, no, it doesn't work in mouse.
Yeah,
no thesis project.
But I don't know that we didn't predict that it would in mouse.
Right.
I mean, we know that there seems to be a difference in a different organism doing something like this in mouse. Perhaps, there's coevolution.
Mm-hmm.
I don't expect that it would have worked in mouse.
Yeah.
Agreed,
yeah.
It is frustrating, though, because it limits what you can ask mechanistically.
Yeah, It does. I mean, the human studies and babies are just obviously challenging. So,
and this was, like you said, Steph, a long term study because they were looking for allergies that developed in children years after they had collected poop samples and mammary milk samples from the moms at, around birth up to 1 year old. And so those allergies and other diseases take years and years to develop. So this is retrospectively looking back and then looking for those correlations based on these outcomes years later, which is interesting. Right. And you can criticize they did or didn't have these samples, but they had what they had at the time and they were able to bank all of the stuff and find this stuff, which is really amazing.
Yeah. And I believe it's Megan Azad who is the PI of the cohort. I've seen her publish different work, and I think bravo to the Canadian government for funding this, because these are questions when I get asked about breast milk immunology and do they correlate. A lot of the data is there, but it's loose. It's hard to really grab a hold of. And that's because the studies are just challenging to get the numbers, to get the statistical power. Yeah.
And I looked it up. So this, we've been saying child study and it was capitalized in all caps in the paper. And I was like, was that an acronym? Like it is, but it's very cute, right? It's a child study, but it's the Canadian Health Infant Longitudinal Development Study. So yeah. So
you always have to have a cute acronym for your cohort to get funding.
So that in the discussion, they say, understanding how milk promotes health and protects against disease is important because we could design milk alternatives or promote healthy milk production, right? Because not all milk is equal, I suppose. So that would be interesting, right?
Yeah, and I think when I talk about milk research and milk immunology, it is you have to be very sensitive about how you're describing this, because you would not want the message to be that mom deciding to not breastfeed her baby and get formula caused her baby to have allergies. Yep. Obviously, we have created a scenario where most babies in the West, in developed countries, are surviving. One of them reasons is ample access to milk alternatives. And that, though, could cause things like allergy issues, maybe lack of protection against respiratory infections. And so you're trying to say, okay, there is a benefit. You didn't cause this in your baby, but how can we offer you a substitute based on science that would help the child? Right. So it's a very nuanced discussion when it comes to.
I always think I have a friend whose daughter had some nutritional issues when she was born and couldn't drink breast milk. And so when I think about the substitutes that she had to have that had the right nutrition, you could imagine supplementing that with something that could do this or something like that. And so there are a variety of situations where breastfeeding isn't an option,
right,
for whatever reason. And it's important to make sure that you can still kind of allow these positive benefits to happen no matter what.
But I think this helps. When I talk to lactation consultants, there are kind of these deciding moments, and a paper like this that they could take to their client and say there are clear benefits of breastfeeding might help somebody make that decision to breastfeed.
I think, though, we have to keep in mind that breastfeeding is a privilege in some ways, in our country particularly, because parents are not given enough time off to bond and to develop an appropriate breastfeeding connection. And then also when they have to go back to work, many don't have access and support to be able to pump, to be able to collect the breast milk, to continue to feed it to the baby. So I don't think we should hold those who can at fault. It's nice if the mom can stay home for 6 months. And in some countries, that's the norm.
Right.
In the US, it's not the norm. It's 6 weeks.
Right. If I mean, gosh, service industry, they would just not be able to make money during that time. It's not a paid time off. I totally agree, Cindy. It's a real United States problem, this conundrum. Yeah.
But if we want to make babies healthy, we need to give them breast milk.
Yep.
Yeah.
So
it's good stuff. If we could make it, that would be great, right?
If we could identify, I'm envisioning a way that you could add a monoclonal to formula that targets this bacteria.
I guess I worry a little bit. I worry a little bit about that because we don't know what else is in there in combination. Yeah, and just knocking down one bug in the absence of any other manipulations might not turn out the way we think it is going to.
Sure.
So I think it's hard to know whether that's the right approach or not.
Right. And it's frustrating that this can't be tested in mice. And so this also might be a situation where we need to find out if there are other animals that might have the same organism where we could do some of these experiments.
Yeah. Because if you look at the species associations, E. ramosum is on top, but it's closely followed by a bunch of others, and you may have to give those as well, right? Just a continuum, a line going so lower and lower and lower throughout all these bacteria. And so where do you stop? It's hard to know. Well, it's never black and white, right?
No.
Right? That's not the way biology is. So they point out a lot of similarities between SFB and E. ramosum, but then they say, we do not propose E. ramosum is a biological equivalent to SFB. Right. However, it looks pretty similar. And it's just too bad we can't do E. ramosum experiments in mice. But I think it's quite interesting.
I wonder what it would take, though, because a lot of the ways, so the immunologists approach this, if you have a system that doesn't work in the mouse, you start genetically manipulating the mouse until you get a system that does work. So they humanize mice to certain degrees. If they were able to identify a binding protein that allowed the E. ramosum to bind to the epithelium and drive this response, you could humanize the mouse at that locus and see whether that then supported the binding of E. ramosum, and then you could use that potentially as a model to study it in a little bit more detail.
One thing I didn't point out is that the members of this cohort vary greatly in the properties of their secretory IgA, as you might expect, right? The targeting of bacteria varies 30-fold between the mothers, right? I mean, the levels are similar, but the composition is very different. Yeah. And so if we could understand why that is, that could help also. Maybe something in your diet is all that it takes. Who knows, right?
Or, I mean, when you sequence the bacteria of just interperson variation of just the gut microbiome, that probably accounts for the variation in the baby, because those are the B cells that are trafficking. Yeah.
My favorite part is figure 1A, where they have a milk bottle with antibodies.
I know. That is very cute.
Yeah, I have to use that for the so thumbnail. That would be great.
I love it.
Did you guys like, though, how they showed the ELISAs, the way they I was like, is that how it actually works?
What are we talking? Okay, where
do we have a lot of? So
1B?
It's showing, for example, total IgA, and then your milk sample, or a bacteria and your milk sample. So the IgA is double-sided. Sort of. That's not really the structure when you look at it. But most of the antibodies that we make to other antibodies are to the constant region.
Yeah, it's to the
Yeah.
Fc.
Yes.
It's not going to bind
and so the way
Fab.
they show it is they are showing the parts that bind to the antigen binding to the other antibody. So like the Y part, the tips of the Y binding to the other tips of the Y. And that's not really
Unless
usually
it's
the
an
way analysis work.
idiotypic antibody.
It is.
Yeah, true.
But
Why would you make an anti-idiotypic
No,
when you don't even know what it is yet?
yeah.
Right.
Yeah,
So I'm wondering who designed that figure for this paper. Maybe they weren't the authors necessarily.
I don't know.
But yeah, interesting. But I did like the bottle with the little antibodies in it. That was cute.
Yeah, that's very cute. All right, that'll do it for Immune 96. If you have any questions or comments, send them to immune@microbe.tv. And if you like these programs, we'd love your support. Microbe.tv/contribute. Cindy Leifer is at Cornell University. Cindy Leifer on Blue Sky. Thanks, Cindy.
Thank you.
Steph Langle is at Case Western Reserve University. Stephanie Langle on Blue Sky. Thank you, Steph.
Thank you. This is fun.
Brianne Barker's at Drew University. BioProf Barker on Blue Sky. Thank you, Brianne.
Thanks. I learned a lot.
And Vincent Racaniello, you can find me at microbe.tv or Microbe TV on Blue Sky. Music on Immune is by Tatami. Thanks for listening to Immune, the podcast that's infectious. We'll be back next month.